And we will move on to the next speaker, who is Dr. Signe Altmäe, currently at the University of Granada as a Ramonica Hall speaker. Sorry, as a Ramonica Ika Hall postdoc, and she graduated from and got her PhD from the University of Tartu in Estonia. And you may think that this is a curious career path, and it is, but for the opposite reasons. You think it's much more likely to end up in Estonia than to come out from Estonia, since Estonia is really a powerhouse of biomedical research in Northern Europe. I know of a number of PhD graduates who have ended up with their postdocs in Tartu, so it's really an impressive place. And Dr. Altmäe is going to talk to us about the funny situation. You know, these little tadpoles in semen may have unwanted company in the form of microbes, or maybe, or at least she's going to explore their presence and a role for them. So, Signe, whenever we are ready to share your screen or your presentation.
Good morning for everybody. Thank you for the nice introduction. I can just clarify. I mean, of course, research is my interest, but I'm in Spain basically because of love. So love moves the borders. And yeah, I did a big part of my studies and research in Sweden, in the Karolinska Institute. And there I met a very nice guy from Almeria, and here we are. So, but yes, Estonia is doing very well in research, and I'm always very proud to say I am from there, and I still keep collaborations. Okay, so I'm using first time this platform, so I don't know how can I share my screen.
The easiest thing is that you go to the purple tab on the bottom to the right.
Yes.
Click on the third icon. It's something like at the bottom you'll see now 3 icons: one for the chat, one for the people, one for something like sharing.
Share content.
Share content, yeah.
Share content.
And then
share.
The second option, share the application or the screen. That should do the trick.
Okay. Is it all fine now?
Yeah. Okay. It's perfect. So Signe, the floor is yours.
Okay. So yeah, the topic is the seminal human microbiome in health and disease. So before I go into the semen, I would like to clarify. We all talk about microbes, we all talk about bacteria, we all talk about microbiome, microbiota, but it's often confusing what is what. So just to clarify what we mean by microbiota and microbiome, what is the difference? The microbiota is all microbes, all bacteria, all viruses, archaea, fungi that live in or on our bodies. And microbiome is, let me see, yes, I can change. Microbiome is the genetic material of it, if we make it simple classification. So if we talk about the experimental studies where the sequences of microbes have been studied, so the correct way to talk is then that we are studying microbiome. And if we talk about alive bacteria, all that are inhabiting our bodies, then we use the term microbiota. And it's the same here, repetition. So microbiome, the entire habitat that includes the microorganisms, the genomes. So basically, if you remember that microbiome, we refer to the genomes, With this, we are fine. And the microbiota is the community of microorganisms present in the environment. And as I mentioned in the previous slide, that it includes bacteria, viruses, fungi, archaea, and the other microeukaryota. But still today, in different levels of research, when we talk about microbiome or microbiota, we mean actually bacteria, because this is the main component that we are studying today. And if you are confused which one is correct to use and you don't want to make errors, you can always just mention microbes or microenvironment or microorganisms. So with this, you are a bit in a safe side. You are not making mistake with the microbiome or microbiota. And yes, of course, with the research, with the time, we are now changing the concept that the bacteria are not all bad. That was before. Virus, bacteria in general connects to something that is bad or harmful for our bodies, which is not true. Basically the opposite. So, and what makes it complex, all the research of the microbiome, is that before we thought the genome was complex, but now looking back, it looks like it's actually much more easier to understand. All humans are 99% similar, and they share around, we have around 20,000 genes, and the genetic material from the mother and father is passed to half and half to the child. But the reality of the microbes on our bodies is this. There is, of course, some inheritance from the parents, but finally is the environment where we live influences it much more, what we are eating, and etc. So what is the result is that we are only 5% similar between individuals. And there is a bit more microbes in our body than our cells ourselves, different species of bacteria, and 100 times more genes, bacterial genes or microorganismal genes, than our own genes. So that's why it's even a concept of second genome has been coined. And we don't know which one is ruling which one, who is driving whom. And this again, with the previous slide, that we are 5% similar in the patterns, composition of the microbes in our body. It's just illustration that the same sites between different people, healthy people, and you can see that the composition of the microbes is totally different, and they are both healthy. So what I think what we have to think or change our concept is that it's not so much about specific bacteria. I mean, we are not talking here about infections. This is another story. But talking about the microbes, microbiome, microbiota in general. The concept to change is that there is no one or single or two or three bacteria or viruses that are changing or causing disease. It's more the imbalance in the homeostasis. So it can be different amount of, let's talk here about the case of bacteria, good or bad. And finally, what leads to diseases or complications in health is the imbalance of the content or composition of the microbes. And yes, is that you think that you are home alone, so that just to know that you are never alone in our life, knowing how many bacteria we have. Why it's been recent years has been a big boost of the microbiome research, and it's coming from every aspect. You can see studies publishing about microbes, microbiome, in health and in disease. And it has been boosted with a big project funded by NIH, Human Microbiome Project, which was launched in 2007. And the idea was to identify and characterize human microbial composition. They have a lot of information in the webpage if somebody is interested in searching more information. But what I wanted to highlight with this Human Microbiome Project is that more than 10 years ago now, they published that our bodies truly, they were showing microbes. And that, of course, the biggest part of it is in the gut. Also in the mouth, a big part, the airways, but interesting urogenital tract. It was most the microbes detected belong to this site. So now we are focusing on this area. And before we are going into the semen, seminal microbes, I would just like to highlight different techniques to study the microbes. So the most common, the most used in today is the 16S RNA gene sequencing. So, I mean, like with any other analysis, you can go in different levels. So this is you can analyze the microbes on DNA level, on RNA level. Also, they are producing proteins and metabolites. So the easiest and the most common is to analyze the bacterial content in DNA level. So we are just focusing on a gene that does not exist in human. It's the bacterial gene. And based on this sequencing of this gene or specific area of the gene, we can detect which bacteria we have in the sample. Of course, now when widely used, and is being used more and more, is the whole genome sequencing of the bacteria, not only targeting one gene, but sequencing all genomes of the bacteria. Is more costly and has technically is more challenging, especially when you're dealing with the samples where you have very low microbial biomass. The next level is to analyze in RNA level, which means already that that means that to detect the transcripts of bacteria, that means that these bacteria are alive, functionally active. Because when you analyze DNA level, you don't know if the bacteria is dead, if it's just a transcript or a sequence of DNA, because just one sequence of DNA can survive thousands of years. So this is the problem, because if we're analyzing this, we don't know if the bacteria are alive or not. And then proteins and metabolites. These are another area of a challenging sector. And of course, it's the future to come, but I will explain a bit later why this part is challenging. Okay, so now we're going to the part of the semen, seminal microbiome, and all studies that will be presented today concluded they are based on the 16S RNA sequencing. Until now, there is only one study that has done in semen the whole genome sequencing. So it's published this year and it's very fresh. So we are focusing on this part where all the previous studies have been performed. So the semen, and it's just an illustration here that electron scanning microscopy, that here it shows that some bacteria are able to attach to the sperm. Okay, semen. It's composed of a mixture of fluids and sperm cells, contains different proteins, different lipids, saccharides, vitamins, minerals. The pH is very good for bacteria to live. And so basically, looking at this data, semen is a perfect place for bacteria to thrive. And it is so. There have been now 10 years of research in this field, and surprisingly, there are not so many studies. I'm saying surprisingly because comparing to other research areas, for example, in the female reproductive tract, when you need to take a sample from the uterus, first you have a limitation of getting the sample, then the amount of sample is limited, and to obtain semen is relatively accessible. So that's why it is a sad truth that this field of research is lagging behind other fields. So around 10 years of research in detecting different microbes inside the semen, and less than 15 studies have been published so far in the topic. So, but the studies that have been published, what are they showing? That there is plenty of bacteria inside the semen. And the first study that was published in 2009, it showed that there was actually inside the semen, there were more bacteria than spermatozoa. And different phyla, it's just a higher level of grouping. And so where does it come from? How come we have we find the bacteria inside the semen? So there are different routes, different possibilities. It can be, of course, from urine, urethra, coronal sulcus, vagina, mouth, gut, and blood. These are different ways. And the truth is, today we actually don't know exactly where does the microbes come. But what has been done is that has been analyzed the similarities between the semen and urine in the context of bacteria. And surprisingly, here you can see, okay, here was also a rectal sample, but what we are interested now is this column of seminal samples and urine samples. And here are every color represents different genera of the bacteria. And you can see there are similarities. And if you are looking into the numbers, then the similarity is 30%. And the same number has been also obtained by one older study that was based on the culture-based methods. So it is actually surprising that only 30% of the microbial content in the semen is shared with urine. So where does the other 70% come from is the question. We summarized the studies a couple of years ago now that were done by that moment. And so the hypothesis is that if 30% are shared between the urine and the semen, then the big part might originate from the upper reproductive tract. Okay, so we were looking what has been done, and there has been published one study in the testes, one study in seminal vesicles where they analyzed the microbiome, and it was done in mice, not in human, and a few studies in prostate. And indeed, if we look here on the phyla level, the same color represents the same phyla. So big part is Firmicutes, Proteobacteria, Actinobacteria, and Bacteroidetes. So this is the composition of the seminal microbiome, and we can see that they do share quite some similarities. But of course, we cannot make strong conclusions because the testes, this study is based only on one study, these results, and the study, of course, what happens with these more invasive samples, you cannot get the samples from healthy controls. So what we are comparing or analyzing is disease versus non-disease, but that doesn't mean it's healthy. For example, in the prostata has been studies sampling of the site of the prostata cancer and the other side of the prostata that didn't seem to have cancer. but we cannot rule out that already the microenvironment is changed, is not comparable with the healthy. So what is all this part is actually lacking is healthy controls, and I don't know how it's possible to get ethically, or even how to get men to volunteer to participate in this kind of sampling. So this is a big challenge. So what we also wanted to investigate a bit further this topic. So what we did is we took a single picked testicle spermatozoa from 10 different patients, and we analyzed the 16S sequencing, RNA gene, and we had also, in every step, we had a set of negative controls, and the results, what we got from comparing the different patients, provided different cells of different maturation phase of the testicular spermatozoa. And what we saw was that with the red here, you can see is a contamination. And blue was so-called true of the detected reads of the bacteria. And here is, you can see also, is the column is the samples, what kind of bacteria we detected, and this what bacteria we detected in the negative controls is basically identical. No, I mean some slight differences, but what you can see from here is that the big part of the experiments we performed, the samples we had actually had a contamination inside. And it's another way of presenting the results. With the dots are the bacteria we detected in our sample, and the bigger the dot, the more amount of the bacteria or reads we detected. And the negative controls have quite similar pattern. There was some few exceptions that we didn't detect any of these bacteria in the negative controls. So what is the message of these experiments is that we have to be very careful when designing microbiome studies, and especially in the case of low microbial biomass, because all the tissues inside our bodies have rather limited amount of microbes. I believe they are not sterile, but the amount, is it detectable, or what are we detecting when we are doing these experiments? Is a true reads, or is the contamination that can come in any step? And now is a beautiful review published recently, and they concluded, they summarized seminal microbiome studies, and here what they are seeing. This was the testis part, and this is the semen part. And what they are highlighting is that with the blue, the bacteria with the blue had positive associations, positive values with morphology, sperm count, with normal semen analysis, motility, and the orange ones, they had like dysregulated or imbalanced environment, and these bacteria were associated with negative values in the sperm analysis. And of course, there has been associations of the microbes with DNA fragmentation, fertilization ability, and IVF outcomes. But so these studies are based on based on 10 or 9 studies in the field, and the problem is that all the studies have used, they are hardly comparable. So this, I would call it this slide like the first introduction or potential showing where we could go, where are the ways, which areas or aspects the microbes could influence. But I would not develop yet any any treatment options or any tests based on these results, because I would call them rather preliminary. And also, there are different hypotheses that the seminal microbiome influence male can influence also couple health and the offspring. These are all hypotheses. It's not been shown, but what has been shown is that the couples share this called seminovaginal microbiome. They do share in active sexual life. They share the composition, microbial composition, and the other is that also has been shown that young men who have never had sex before, compared to young men who have had sex, that the microbial composition in semen is less rich and there is less amount of microbes. So clearly there is interaction. And what I would like to stress here is that the studies that analyzing female microbiome or male microbiome idealistically should also take into account the couple microbiome. I know there are no ideal studies, but if somebody has to design a study, I would recommend to consider the partner option of the partner also to study. And what adds more to the complexity of the microbiome studies is that there are so many factors that can influence the composition. And it's almost impossible, in my opinion, to control for all of these factors. It's just illustration that age, ethnicity, location, hygiene habits, sexual habits, drugs, nutrition, stress, sport, all these have been shown to influence the composition of microbes. So why I was saying before that I believe that the results are rather preliminary, and I mean, clearly more research is needed because the studies performed are in a relatively small sample size. There is often lack of control group or no control group, a lack of paired urine sample, because as I mentioned before, they are sharing 30% of the similarity, urine and sperm, or semen. So when you're analyzing semen, seminal samples, you should also analyze the urine samples, just for controlling, if not further interest. There is in the experiments, there is lack of use of positive and negative controls. Actually, until recently, there was only one study that used negative controls throughout the experiments. And we know that we need to have positive controls and negative controls in all steps, already from the moment of obtaining the samples. Then there is a term of ketome in laboratory reagents. And what is the ketome? It's just has been shown in different studies that the kits, laboratory kits that we are using, and often are based on produced from different bacteria, enzymes from different bacteria, they contain sequences of bacteria, so they are not sterile. So this already can influence the results. Then why it's very hard to compare the studies is because they all are using different protocols, different analysis platform, etc. And as I mentioned, it's almost impossible to control for all lifestyle factors, or almost to any. So if anybody is interested, I have put here two references recently published that the minimal standard requirements are proposed. So how microbiota, so okay, we have in the semen, we have microbes, and so what are they doing? The truth is, we don't know exactly. We can guess based on the studies in the gut that there are good and bad bacteria, and there is always competition between the bacteria to inhabit the niche for living. So there are different sensing receptors or leukocytes to detect which bacteria we have, and if they are bad or beneficial or harmful, the different cascades defense responses are activated. And the last part of the presentation where I want to reach is that we have to think that we are a human, no? And we have a lot of different bacteria in and on our body. So there is always a crosstalk with between the host and the microbes. So how is the crosstalk obtained? Because we are the metabolites, microbes that are producing when they are living with metabolism or the metabolites, they can influence our bodies. And we need them, for example, vitamins and long and short chain fatty acids and more. So when we are analyzing, we want to understand the role, the function of microbes in semen. We should also start to see which kind of metabolites these microbes are producing and how they could affect the human body, or in this case, a male reproductive tract. So what we did, it's very fresh, actually, it's the first time I'm presenting the results, but very briefly, we analyzed 100 men, the seminal samples of 100 men, and we did mass spectrophometry, spectroscopy analysis, like untargeted. We wanted to detect all kinds of metabolites that are inside the semen. And what we got? We detected almost 700 different metabolites in the semen. And there are DHA, DPA, CMP, choline phosphate, names that we have heard here and there. And so now what we are, and then the next what we did was that we compared the azoospermic and normospermic men, and a lot of metabolites that were differentially present in the two study groups belong to the lipid metabolome pathway. So what I want to highlight with this, we analyzed the metabolites in the samples. But now the problem is which one are human ones and which one are the bacterial ones. And this is what I said before, I want to reach here back, because the genetic code is universal. I mean, the DNA sequence, yes, is specific. We can detect different genes that we don't have, a human don't have. We focus directly to the bacterial genes. We analyze bacterial genome that humans don't have. Then we produce the transcripts based of the sequences, and we can still distinguish are these sequences bacterial or human origin, with some exceptions, but yes. But now from RNA to produce proteins, the genetic code is universal. So we produce, based on the same sequence, we produce the same proteins, and the metabolites are the same in all organisms. So what we are facing now, the difficulty is, of course, it would be the best to know which kind of metabolites are inside the semen, and we go directly, we could even detect biomarkers, we could directly, okay, these metabolites, these are from this bacteria, and maybe are associated with sperm motility or sperm reduced quality, sperm count, whatever. But now is the complex part is to know which belongs to human and which belongs to the bacteria. So to conclude all the presentation, we end up with more questions than answers. The knowledge of the seminal microbiome is in its infancy. The core microbial composition in health and disease is actually not established and needs to be confirmed in many studies in different ethnicities in a bigger sample size. When we're analyzing DNA sequence, we cannot guarantee that this DNA sequence means a live bacteria. We don't know if the microbes are residents that we are detecting, or the tourists, or are they invaders. As I mentioned, that the DNA sequence can remain even after infection, for example, hundreds of years. So we don't know what we are detecting. Is it alive? Is it not alive? Is it a resident, or it was an invader? And when we are detecting that this bacteria could be associated to this disease or this parameter in sperm, we don't know, is it the cause or is the consequence? And finally, if we detect the dysbiotic condition of semen, can we treat it? And if yes, with what? So these are, I end up the presentation with many questions, and I would like to acknowledge the research team that we are very enthusiastically and happily working together, and the funders. Thank you very much.
Thank you, Signe, for this interesting presentation. I'd like to ask everyone that has a question to wait for the end of the presentation, so us know so we don't have these distractions in between. And we've had another an example in right now that Paco Freire had a question, but then in the middle of your presentation, but then you sort of asked it in your following slides. So it's so please everyone wait for the end of the presentation before sending your questions to the chat. And so please do it now. Any questions that for Dr. Altmäe you can. Okay, so we have one. From Sai Kamal, who thanks you for your presentation and wonders whether we talk about IVF, ICSI, we process the semen, and studying chance we alter the microbiome of the semen in these ART techniques. Is this affecting the ART results? What would be your opinion on that?
I know that there has been studies that they show that the ART conditions are not sterile. This has been shown from a group from Estonia, actually. But if they are negatively affecting, I think we don't have these answers today. Let me see, what is the question again? The effect if it's a direct interaction with semen. There has been, but they are experimental. I mean, they are not in vivo, they are in vitro experiments. And has been shown, yes, that if the bacteria attaches to the sperm, it can damage the sperm motility. But it has been done only in the laboratory conditions. And I don't remember which bacteria they used for these experiments.
More questions for Dr. Altmäe? Yeah, if I might go on then, and questions that I had, but you also addressed is whether, for instance, lifestyle may create or other factors may create spiritual associations between the microbiome and the sperm quality or functionality. And
yes,
and how can we resolve that?
This is a question maybe you should ask in 5 years time from now. How can we? But it's true. I mean, the studies that have been published so far, they are not even reporting well the lifestyle factors. I mean, you have to take into account BMI, nutrition, but you know, the nutrition is very complex. It's how to take into account all of this. I think it's a big challenge we are facing in the. Is a big limitation of today's experiments, and how to modify, how to, I mean, what are these pages for people telling internet how to improve fertility, eat well, no, eat healthy food, and these things. So this, I believe it, actually, I start to believe now that there is some truth behind. I didn't believe in this before, but now I start to, I mean, has been studies in mice showing that if you are eating nutrition with high-fat diet or low-fat diet, you can modify the quality of the sperm.
And so you think that part of this modification goes through the interaction between the diet and the microbiome?
Yes, because there seems to be also interaction in the gut microbes. And the seminal. I mean, it's the whole body in the end, and the bloodstream, and the mouth. And I mean, the truth is we don't know even exactly where the microbes come from, what are inside there.
Okay, so we have
I see here a question.
Yeah.
So asking whether the microbiome is a sign or a cause of infertility is a possible translation to knowledge to the art field. We are still in the field of infancy. Do we have an option based on other organs fields on the relationship between the two? Yes, I mean, it's hard to answer this. For example, we are doing a lot of research in the female reproductive field, and what we are facing now is that when we take the samples, we can detect microbes in the uterine samples, endometrial samples, and there are correlations that specifically in this case, more lactobacillus is more beneficial. You get better implantation rates. I believe in some ways it also reflects the vaginal condition, because the vagina should be lactobacillus dominant. If the imbalance is there, you have other issues. So, so I think it would work like a biomarker. It does work. It seems to work in female testing in the field of assisted reproduction. But if we want to treat the women based on the sequences, what we detect, I think it's too early, because it's just the sequences, and they are not. We cannot be sure that these are alive. And if you're treating the patient with antibiotics, you are killing the live bacteria. You are not killing the DNA sequences. So I think there is still like a gap between the technology, what can detect DNA sequences, fine. Maybe it can be a biomarker, but in my opinion, it's not enough yet to treat something that we are assuming is alive. So this is the case of the female. In the male, I mean, there are basically 10, 11, 12 studies that has been showing the different microbes. They are not even 100% confirming each other. So I think we are even far from even suggesting some biomarkers or. So basically, I think I'm not really answering the question, but.
So, Signe, thank you very much for your presentation. We need to move on to the next presentation. So again, thank you very much.